Okay, thank you for attending and good day for all of you. So I just want to give you a short talk about the recent finding that we had, and the title of my talk is Multimodal Analysis of Circulating Cell-Free RNA, Circulating Cell-Free DNA, Genomic DNA, and Circulating Tumor Cells from blood samples in cancer research. So my name is Thorsten Voss, and I'm a research scientist in the PreAnalytiX R&D department from the joint venture PreAnalytiX between QIAGEN and Becton Dickinson. So we have in the next slide a short legal disclaimer, so that every product that you will see here in the next slides is for research use only. And now I come to the content of my talk. So I divided the talk in 3 parts. So the first part will be an introduction in this field. What are the main topics in the field of liquid biopsies? And I start with the specific circumstances about ccfDNA and what's beyond of this. And then in the next 2 parts of the talk, I will give you a short summary of 2 very recent studies that we have done, where we focused on the multimodality and analysis from liquid biopsies. So in the first part, we look at a study where we looked on ccfRNA, ccfDNA, and gDNA from the same single blood sample. And in this final part, I will show you a study where we expanded this workflow to CTC as well. So for introduction, so in this first introductory slide, I start from the beginning. Many of you probably also already know this, but to have everyone on the same page, I just want to show you here this scheme of a blood vessel with a tumor adjacent and connected to it. And we just want to remind you that we have mainly 3 processes which are important for the liquid biopsy from blood. And these 3 processes are shown on the left of this slide, which releases, or which are the reason why biomolecules are released from the tumor into the bloodstream. So in the middle part, you see a scheme for apoptosis. So apoptosis, so the regular cell death, is of course the main process that is involved in releasing compounds into the blood cells. So apoptotic processes, of course, occur in tumors as well as in every other part of the body. And during this regulated cell death, you get these regulated process of degrading genomic DNA as well as the rest of the cell. And via this, you get defined fragments of the genomic DNA, the so-called nucleosomal fragments from the cell released into the blood. So these nucleosomes shown in the blood vessel on the lower part contain ccfDNA, so circulating cell-free DNA, which is wrapped around the histones and normally in this mononucleosomal fragment of a length between 160 and 180 base pairs. So this is the main part of the ccfDNA, but the apoptosis processes also release some particles which are surrounded by membranes, the so-called exosomes or nuclear or other extra particles, which are also released via this apoptotic process. Besides apoptosis, we have also 2 other mechanisms releasing biomolecules into the bloodstream. The upper one is the necrosis. This is of course important, especially for tumor which releasing their compounds into the bloodstream. So this unregulated process is rapid dying of the cell, and via this process you get other fragments of DNA into the bloodstream, which are of different lengths, and but also contain the information from the tumor. The third process that I want to mention here is the secretion process, where the cell is not dying but secreting actively some compounds into the bloodstream, mainly extra vesicles, which are mainly called exosomes, and these exosomes mainly contain circulating cell-free RNA. So we have these three main compounds of liquid biopsy molecules that are of interest. So the ccfDNA, as I said, a part mainly of this mononucleosomal feature, and then ccfRNA, which is mainly found in the exosomes. And then, as another important compound, of course, we have the release of complete cells from the tumor into the bloodstream, which are called CTCs, so circulating tumor cells. Coming from this overview, I just want to show you why it is so important that we have some kind of a standardized workflow and some kind of stabilization, which is strongly needed if we work with ccfDNA as well, with the other molecules I mentioned and the CTCs as well. So what you see on this slide here is a picture from an electropherogram from an Agilent bioanalyzer, where we look at ccfDNA isolated from plasma from an EDTA tube, where the blood cells are directly separated from the plasma directly after the blood draw. And you see here the normal pattern of ccfDNA for a healthy subject, with a main peak of ccfDNA in this range, as I mentioned before, of 160 to 170 base pairs. And some other smaller bands here for multimeres of this mononucleosomal DNA, as well as some fragments of larger DNA next to the upper marker of this assay on the right. So this is a picture what you see as it should look like for ccfDNA from healthy patients, as I said. But this looks completely different, which is shown in this slide here, when you store the blood for longer times. And what we have done here, we have a replicate sample here, also collected in ETA, and stored it for 6 days at room temperature. And what happens then is that you have an increase, massive increase of gDNA release and also apoptotic band release from the dying white blood cells, which is shown in this darker bluish graph that is overlaid here for the original one. And what you see here, that you have a huge release of DNA into the plasma, which covers the original ccfDNA and overlay it so that it is difficult to get the original signal and find your target molecule in this huge background. So therefore, it's very important that you have a stabilization of the ccfDNA, especially the stabilization of the blood cells, so that they cannot release their DNA. So we, and as well as other companies, developed a solution for this, so a specialized blood collection tube. Here in our case, it's called the PAXgene Blood ccfDNA, a tube where you see this tube on the right. This has a stabilization solution in it, where you get a direct stabilization of the cells with the blood draw, so that it is prevented these cells to release their DNA into the plasma and overlay the original ccfDNA. So, as I said, this stabilization solution prevents the apoptosis of the white blood cells, but this also prevents the apoptosis and the disintegration of the red blood cells and the hemolysis, which is not that important because of the DNA, because the red blood cells, of course, do not contain DNA, but the red color of the plasma, of course, has some other negative impacts of the plasma harvesting, and as well as some downstreams can also be impacted by a huge hemolysis. Furthermore, this tube has also this non-crosslinking feature of their stabilization solution, in contrast to many others. So we have no modifications on the ccfDNA with this tube, and therefore it's compatible with very sensitive downstream methods, as I will show you in the next slides, especially also with methylation-based assays. Further feature that are also important to know is that this tube has a plastic tube, so you have a minimize of risk of breakage, as well as you have this hemoguard closure, which prevents splattering of the blood when you open the tube, and this of course enhances the safety of your personnel in the lab. So coming back to the slide that I showed you before, so if we use such a tube, so in this case our PAXgene Blood ccfDNA for blood collection, as you see it here in this graph, we expanded the experiment by 2 tubes, one PAXgene tube that was where the plasma was directly removed after blood draw from the cells, and one where we stored the blood for 6 days, similar to the EDTA, and then do this removal and ccfDNA isolation. And what you can see here is that the blood collection tube, the PAXgene tube, does its job. So you see no release for this bluish graphs here of the DNA into the plasma. So the white blood cells are preserved by the stabilization solution. If we expand this experiment further, for example, with another tube from the company's track, which is also very common in the field and widely used, which also has a solution in it which prevents the white blood cell for lysis. So you see also from this here in this greenish color here, the graph that after 6 days you don't see a massive release of genomic DNA into the plasma. So also this tube is doing its primary job. So no release of genomic DNA. But what you also see, if you look a little bit closer to the main ccfDNA peak here, you see that the peak of the ccfDNA from the track tube after 6 days is a little brighter, and also you see a small shift in the size of the DNA, which is also shown in the table on the right hand of this slide, where you see that we have a slight shift of the DNA in size measured by the Agilent bioanalyzer, and this is because the track tube using a different methodology, different compound in the stabilization solution. So this tube contains formaldehyde-releasing substances, and this formaldehyde crosslinks the cell membranes and therefore prevents the gDNA to be released from the cells. So this is obviously very effective, as you see from the slide, but on the opposite, which is the downside of this, is that this crosslinking is of course not a directed process. It crosslinks everything that is in the blood, and therefore also some of the ccfDNA molecules are crosslinked to other cell compounds and plasma compounds, which is seen by this shift. And these crosslinks cannot be completely released from the prep. So you can say, okay, so this is a nice artifact, and does this anything mean for my downstream application? And so we can see in the next slide that this is the case, and this is important to know for you. So what you see here in this small study that we have done is an experiment where we collected blood from healthy donors in 3 different tube types, so the PAXgene tube, the track tube from the slide before, and another third tube from a different competitor, Biography, which has a similar strategy as a track tube to preventing the gDNA release. So, and we collected blood from healthy donors in these 3 tubes, and then directly after blood collection, we spiked in sheared DNA, which was sheared in the size of ccfDNA, so 170 base pairs. And this DNA that we spiked in carries mutations of the EGFR gene. So these 3 very prominent mutations was in this DNA, and we spiked in a very low number of copies, so near the limit of detection of the assay that we used for detection. And after 7 days at 30 degrees incubation, we removed the plasma from the blood cells and isolated ccfDNA, and then have done an assay which can detect these 3 mutations, so qPCR assay, and looked what are the results. And what you see here in these 3 different colors, you see this grayish, dark gray color. These are the samples where we could not detect any of these, or where we could not detect the spiked in mutations. So no signal, no CT for these mutations anymore. And then we have this dark greenish color where we could detect the mutation as it is valid for the assays. So this assay, this component was an assay where you detect the wild type and the mutation and build a delta CT to have a valid reaction. So all these dark green samples have a valid reaction. And then we have also this light green color where we get a CT for the mutant that we spiked in, but this was out of the specification of the assay for this delta CT. So overall, what you see from these after 7 days of incubation, you lose a signal in many of this track as well as the biomarker tubes. It's different between the different assays, but you get for the PAXgene at least the signal, the CT of the different mutations for nearly all of the samples. So you can really see that the chemistry in the tube that you are using can have an impact on your stabilization and your assay outcome. So what I showed you here is an example that we need some stabilization and we need some standardization because of the to prevent errors that can be made from if you work with ccfDNA. And this is also noticed by officials from different countries, and therefore they decided to finance a great consortium which look into the standardization of preanalytic processes, where especially for certain compounds, and this ended up in an ISO norm which was published last year for ccfDNA isolation, and this is shown on the bottom of the slide. And within this ISO norm, there's a clear guidance how to work with ccfDNA, how this needs to be standardized. Okay, so coming to the second part of my talk and to the first study that I want to show you with respect to multimodality. So in this study, we looked into a workflow where besides ccfDNA, we wanted to show and to look whether other analytes from the liquid biopsy, which are important, can be isolated from the same blood sample. So we for the workflow, so we collected blood again in different sample tubes. So we used our PAXgene tube, but also some of our competitor tubes, so the STRAC tube that you have seen in the last slide, as well as the biomarker tube. And we also included the new STRAC RNA tube, which is dedicated to ccfRNA isolation. So we collected blood in these tubes. Then we stored the blood for 3 days at room temperature. Sometimes we also incubated it for 6 days, as you will see in the next slides. And then we centrifuged these tubes according to the ISO standard I mentioned before, or the instructions of the tube provider. Then we isolated the plasma, and from this plasma we prepped the ccfRNA with three different kits. So we worked with the EXORN-easy kit, which is focusing on the exosomal RNA. So we have the isolation of the exosomes and then isolate the RNA. We work with the microRNA-easy kit, which isolates the RNA from the complete plasma exosomes as well as other external RNA. And then we isolated the RNA from the plasma also with the QIAGEN circulating kit, which is capable of isolating the RNA, but as well as you see the circulating cell-free DNA, so that we have the DNA and RNA from the plasma. And then after the plasma prep, we also looked into the remaining cells that you have still in your tube, and we isolated the genomic DNA from the remaining cells as well. And then we analyzed the content and the yield with PCR assays to get an idea what comes out of it. And that is shown in the next slide. So in the first part of this study, we looked into a short time course experiment. We wanted to know how good we could stabilize with the CCF, with our PAXgene ccfDNA tube, the RNA in the plasma compared to EDTA. And what you see in these graphs is on the left 3 microRNA assays where we isolated microRNAs. The left one is one that is mainly found in exosomes, and the right one is the one that is mainly found outside the exosomes in the plasma, and the LED7R in the middle is the one that is found as well in the plasma as in the exosomes. And you see that the behavior is a little bit different between the different microRNAs, but overall you see that you get relatively good stabilization in the PAXgene tube up to 3 days, sometimes also up to 6 days, but always much better than what you get from EDTA. And on the right, you see also an example for mRNA isolation, with mRNAs nearly exclusively found in exosomes, and you also see that the stabilization with the PAXgene tube on the right is very good in contrast to the EDTA. So overall, we can see from this that we get some kind of stabilization for up to 3 days. So we decided to work further with 3-day stabilization. And that is shown on the next slide as well. So in this part of the study, we looked into what is happening with different tubes. So we looked in our tube again, as well as to the 2 Streck tubes, so the DNA, the RNA Streck tube, and the Biomarker tube, and how there we can see how good is the stabilization in these tubes compared to time point 0. And what you see here is, again, you see a small release of RNA into the plasma or in the exosomes after 3 days from the PAXgene tube, but different kinds of release, in most cases higher than the PAXgene tube for the other tubes. So you see it is important what tube you use if you are interested in circulating cell-free RNA. On the right, in this bluish graph, you see that we also looked in, as I said, in the ccfDNA. So from the same plasma, we isolated the ccfDNA, and you see that there we have nearly no change for all the tubes that we looked compared to EDTA, where we have a huge release of DNA, as we have seen in the first part of my talk. Furthermore, and finally for this study, we also looked in the genomic DNA from the remaining cells, and this is also something that was a little bit surprising for us. So we looked into this, and what we see, you get a relatively good yield for the PAXgene tubes after 3 days, and so directly and after 3 days. Also for the Streck DNA tube, a little less for the Streck RNA tube. Okay, so this we were not so surprised because it's an RNA tube. But we see that we have very little yield for the Biomarker tube, and we have not a real explanation why we see this. But furthermore, we looked at the integrity of the DNA, and you see also here on the right graph here, that from day 0 to day 3, we have nearly no change for the PAXgene tube, as well for the Streck DNA tube. We see little decrease in integrity for the RNA Streck tube, but also for the Biomarker tube, there's a strongest decrease in integrity for this tube. Okay, summarizing this first multimodal study, so I showed you that the non-cross-linking technologies for the PAXgene tube enables the isolation of microRNAs as well as cell-free mRNAs and DNA and genomic DNA from the same sample without any issues and with stabilization features. And we have seen that some of the other tubes have negative impact on the ccfDNA stabilization as well as the microRNA and the gDNA. Okay, so coming back to the overview, so and now in the final part of my talk, I like to show you the second study for multimodality. So in this study, we expanded this multimodal workflow further to the analysis of CTCs. So for the workflow, and then for the workflow overview, I don't want to explain what we have done. So we collected again blood in our PAXgene Blood ccfDNA tube, and then we used the ADNA test technology from QIAGEN to isolate CTCs from this. So we spiked in our in the blood from the healthy donors tumor cells in this blood, and then incubated for certain times and temperatures. And then we used antibody-coated magnetic particles, where we then isolate or enrich the CTCs from the blood, isolate them via magnets, and on the upper part of the workflow, we then put it in the ADNA test workflow, lysis these CTC-enriched fraction, and did RT-PCR and looked in the specific targets from these circulating tumor cells. So we could analyze the CTCs, and then from the rest of the tube, after the CTC depletion, we spun the tube, isolated the plasma, and then again isolated the ccfDNA from the cells. We isolated the genomic DNA. To have this in more detail, we look first at the CTC workflow. So as I said, we collected the blood. Then we spiked in 20 cells into this blood in the PAXgene Blood ccfDNA tube. Then we stored this tube either in the fridge for 2 to 8 degrees Celsius or at room temperature. And then we stored this for different times so that we could look at after 3 hours, 24 hours, 48 and 72 hour storage. Then we isolated and enriched the CTCs from the whole blood and put the CTCs then through these ADNA test prostate cancer panel for AR-V7. And then in the next slide we see the results. So on the left we have the cooled storage, and on the right the room temperature storage. And you see the green color here is for the positive test, and the red for negative test. And you see that after 3 hours we could detect for all samples as well as after 24 hours. So there we for all samples were positive, so we could detect the spike in CTCs. And then after 48 and 72 hours we get a few samples where we could not detect the CTCs anymore, but for most of them we still can detect the CTCs that we spiked in. This is true for cool storage as well as room temperature storage, but for cool storage we see a little higher amount of samples which were positive for the CTC, so which we can use. So then we decided that we should use fridge storage in the fridge if we want to have longer storage. And then in the next step we looked into longer time course experiments, so up to 144 hours, what happens there. And you see here on the right that if you store it for even longer times, that then the number of samples where you could not detect the CTCs anymore is increasing. So we decided that the best workflow for our tube, so the PAXgene Blood ccfDNA tube, would be cooled storage for 2 to 8 degrees and then up to 72 hours. So after we established this workflow, we of course looked into what is happening if we do this in a similar way with competitor tubes here. And we again choose the STRAC tube here, STRAC DNA tube, to have a side-by-side comparison. So for both tubes, then we use the same workflow. So we spiked in again 20 cells in both tubes. Then we used the optimized workflow. So as I said, for the PAXgene, we used the cool storage. For STRAC, we used room temperature storage because in the STRAC literature we found that they say that the best storage is at room temperature. So we want to choose the optimized conditions here. Then again, we stored for these different times up to 72 hours. Again, isolated the CTCs from the blood and used the ADNA test prostate cancer panel. Okay, so the results look like this. So on the left you see the PAXgene tube results, on the right the STRAC tube results, and we see again that we could use here, find very good results even after 72 hours. But the new result is here for the STRAC tube that we see we could detect the CTCs after 3 hours storage, but already after 24 hours and also for the longer times, we could not detect the CTCs for any of the STRAC tubes that we are used. So this is obviously not possible for longer storage with this tube. Okay, so summarizing this part of the second study, so we have seen that for the non-crosslinking reagent, the PAXgene tube is compatible with RNA analysis from CTCs with this ADNA test panel, and we propose the best workflow is at 2 to 8 degree storage, and we could detect it more or less completely up to 72 hours. So for the formaldehyde releasing STRAC tube reagent, we saw that this is definitely not compatible with the CTC workflow if you have to store the tube for longer than 3 hours. Okay. So, and then this is the CTC part of the workflow, and then, as I said, so we also looked, as this is a multimodality workflow, we looked also in the other liquid biopsy molecules. So we looked into the plasma on the right here and also on the cells. And first we show you the results for the plasma part. So from the plasma we isolated the ccfDNA. And for this, we used on one hand these greenish samples where we have which was used for the complete workflow, so including the CTC depletion via the ADNA test, and we also used some control samples where we have no CTC depletion done with them, and compared them side by side. And what you see here with respect to the yield, so you see on the y-axis the yield, and on the x-axis the different storage times, and you see that for both sample types you have no significant difference, so you can successfully isolate ccfDNA still after CTC depletion and enrichment from these tubes. And finally, in this last part of this workflow, we also looked into the gDNA, so we isolate gDNA from the remaining cells. And also here you see a similar pattern, samples as control with no CTC depletion, and in green the ones where you depleted the CTCs, and you see that there is no difference with respect to the gDNA yield that you get from these cells, also not with respect to gDNA purity. So concluding this study, I showed you that CTC depletion had no significant impact on ccfDNA yield and in situ stability. So I showed you as well that CTC depletion had no impact on in situ stability on yield of extracted genomic DNA, and that that this non-cross-linking reagent in the PAXgene tube enables the 3-in-1, so to say, simultaneous enrichment of CTCs, and then further ccfDNA isolation and gDNA isolation from the same single blood sample. Okay, coming to the last slide of my talk. So overall, conclusion of my talk and the 3 parts. So I showed you that the analytes in liquid biopsy like ccfDNA, ccfRNA, and CTCs have a strong need for stabilization if you want to analyze them, and that the whole workflow needs standardization. So this is documented, for example, in this new isonorm for ccfDNA isolation. Further isonorms and standards are on the way in this PEDIA consortium that I mentioned before. So I also showed you that the non-crosslink stabilization reagent in the PAXgene ccfDNA tube enables detection of low-abundant target molecules, as I showed you for this tube PCR with the different EGFR mutation, and this ensures that you have a sensitive reaction, sensitive detection of these targets in your downstream reactions. And finally, I could show you with the 2 studies here that the PAXgene Blood ccfDNA tube allows multimodal analysis for ccfDNA as well as ccfRNA, gDNA, and CTCs from a single blood sample. Okay, so with this, I'm at the end of my talk, and thank you for your attention, and happy to answer your questions. Thank you.